Welcome to the Deep Dive. Today, our mission is to give you a massive shortcut into what might be the biggest hidden library of scientific data in the world.
We're talking about the Gene Expression Omnibus, or GEO. You can think of it as this vast digital archive. For decades, scientists have been depositing millions of molecular samples.
From all kinds of experiments, right? Human, mouse.
Everything. Tracking disease responses, genetic manipulations, you name it.
Right. And they did these studies for, very specific narrow reasons, like understanding one particular cancer pathway.
But they almost never analyze for the elephant in the room.
Aging.
Aging. All that data just sitting there. It's a goldmine of information about what speeds up or slows down biological aging.
And it was just waiting for the right tool to come along.
And that tool is finally here. We're doing a deep dive into a really radical new platform called ClockBase Agent. And this AI, it completely changes the economics of discovery.
Wow, so.
By systematically going back and reanalyzing all that archived data using specialized biological aging clocks.
And, this isn't just running a few stats. We're going to explore how these specialized AI agents autonomously process, over 43,000 comparisons.
43,602 intervention control comparisons. A scale that would take human scientists centuries to get through.
And in doing that, they found thousands of age-modifying effects that the original researchers just had. They completely missed them.
It really is the ultimate hack for biological discovery.
Okay, so let's unpack this. And I think we have to start with the scale, because calling this big data feels like a massive understatement.
It is. The platform integrated roughly 2 million human and mouse samples from GEO, all from before the start of 2025. It's an almost unimaginable amount of raw info.
Can you give us a bit of a breakdown of that 2 million? Because it reflects the different types of molecular data they were looking at, right?
Absolutely. So we're talking about 964,083 mouse RNA-seq samples.
RNA sequencing, so that's gene activity.
What the cell is actively doing. Then you have 852,381 human RNA-seq samples and over 230,000 human DNA methylation samples.
And DNA methylation is different. That's more about these chemical tags on the DNA.
Exactly. It's tied to long-term cellular memory, and crucially for us, it's very strongly tied to the aging process itself.
So you have this ocean of data. To make sense of it all, you need some kind of standardized measuring stick, and that's where the 40 aging clocks come in.
Yes.
But why so many? Aren't all aging clocks basically measuring the same thing?
That's a great question, and no, they are absolutely not. ClockBase used over 40 distinct models for a reason. Biological aging isn't just one single process.
It's many processes happening at once.
Exactly. So you have your basic chronological clocks, but the really important ones are the second and third generation clocks. Clocks like Grimmage and PhenoAge.
And what's the practical difference there?
Well, Grimmage and PhenoAge are mostly measuring accumulated damage, mortality risk, but then you have something like Dundon pace.
The pace of aging clock.
Right. It measures the speed at which you're accumulating that damage. By running 40 different models at once, the AI can see if an intervention is consistently slowing down multiple facets of aging, not just hitting one marker. It adds a ton of robustness.
That makes perfect sense. So 2 million samples, 40 clocks. How do you automate the scientific rigor for that? This is where the specialized multi-agent architecture comes in, and it feels like the core innovation.
It absolutely is. The architecture is designed to mimic a team of expert bioinformaticians.
A team, so each agent has its own job.
3 critical specialized roles. First, you have the coding agent, or the analysis executor. Its job is to dive into the raw data, make sense of the metadata, and then generate aging-focused hypotheses.
Like, did this drug affect aging compared to the control?
Precisely. And then it executes the right statistical analyses flawlessly.
Okay, so that's the number cruncher. But biology is messy. You need an interpreter.
And that's the second agent, the biological interpreter and reviewer. Once the stats are done, this agent provides context. It conducts these super fast literature reviews using a system called RAG.
RAG, Retrieval Augmented Generation. Can you explain that simply for us?
Sure. Think of it like this. The AI has already read and indexed over 6,000 published papers on aging.
Wow.
So if the coding agent finds that, say, intervention X slows down aging, the RAG system instantly checks if that's biologically plausible. Does it line up with known mechanisms? Does it contradict something we already know?
So it's like a real-time automated peer review.
Exactly. And finally, you need to make sense of the results. That's the manuscript agent or the scorer. It synthesizes everything and produces a scientific report with a composite score.
To prioritize the most promising findings.
Yes. The higher the score, the more evidence there is that this finding is real and potentially useful.
This is where it gets really important for me, and frankly, why we should trust these discoveries. Public data is notoriously messy. They had to validate this whole system, right?
They did. And the validation is what makes this whole thing so credible. They took 100 random, complex analyses from the agent.
And had humans check its work.
2 independent doctoral-level bioinformaticians. They manually reviewed everything. The result was just astonishing.
What was the number?
The AI agent achieved an overall correctness rate of 99.3%.
99.3% on complex, messy, real-world data.
It means the platform isn't just fast. It's performing at a confirmed expert level. And that validation means we can trust the discoveries that came out of those 43,000 comparisons.
Precisely. Which is a perfect transition to what it actually found.
And what it found tells us something really fundamental about biology itself.
So let's get into those findings. Out of more than 43,000 comparisons, how many actually showed a significant effect on aging?
A significant minority, actually. 5,756 comparisons, which is about 13.2% of the total.
That's already a huge pool of new knowledge. But the really profound insight wasn't just the number of hits, it was the direction they were going in.
Exactly. The analysis revealed this fundamental asymmetry in biology. Interventions are significantly more likely to accelerate aging than to decelerate it.
Which is just a profound observation, isn't it? It's statistically much easier to break something, to throw a wrench in the system, than it is to find the perfect lever to improve it.
It really confirms what a lot of researchers have suspected. Biological systems are highly optimized for survival. And when you disrupt that optimization, whether it's with a disease, an environmental toxin, or a drug, you're far more likely to make things worse, to speed up aging.
Finding an improvement is the rare exception.
It is.
And if we look at the different categories of interventions, which type was most likely to have an effect?
Disease states. They showed the highest significance rate at 24.3%. That was followed by environmental exposures, then drugs, and then genetic interventions.
The fact that disease was number one is actually really important because that works as an internal check on the whole system, doesn't it?
It's a perfect validation. Of course, pathological conditions accelerate biological age. Things like ischemia reperfusion injury or viral infections were highly pro-aging. In that whole disease group, there was an 83% bias toward pro-aging effects.
So if your clocks can reliably tell you that being sick makes you biologically older, then you know your clocks are working.
You know the system is working correctly, yes.
Okay, now let's talk about something with huge implications for future work. Genetic strategy. Did the agents find that it was better to add a gene or to take one away?
The insight here was remarkably clear. Loss-of-function strategies, so that means knocking out or knocking down a specific gene, they systematically outperformed gain-of-function, or overexpression.
So sometimes the best way to live longer is to take the foot off a detrimental gas pedal.
That's the implication. Knockdown interventions showed a 1.85-fold higher proportion of anti-aging effects compared to overexpression. Again, it fits that asymmetry principle. Forcing a cell to overproduce a protein often just disrupts things and causes damage.
It's one thing for an AI to see these patterns, but we need to know the real biological signals. How did these AI-found candidates stack up against, the established longevity databases?
The agreement was extremely high. It really cements the biological plausibility. For genes, they found an 81% directional agreement with the established GenAge database.
And for drugs?
For drugs, it was even better. The 5 compounds that overlapped with the DrugAge database showed 100% directional concordance, and this included the big ones.
Let me guess. Rapamycin, metformin?
Rapamycin, metformin, nicotinamide riboside. The AI basically validated the known superstars of longevity science.
Which is great. It proves the system can find things we already know work. But where's the new discovery potential?
That's the real treasure here. Of all the drug candidates the AI identified, 98.2% were not previously linked to lifespan in the Drudge database.
98%.
It's a massive, untapped reserve of potential new geroprotectors that the agent has now brought to the surface.
That's incredible. But there was one finding about drugs that are already in clinical use that, wow, it's truly alarming. The analysis of FDA-approved drugs.
Yes. This showed a deeply worrying pattern. Among the FDA-approved drugs in the dataset, the ClockBase agent found a really significant imbalance. 136 compounds were identified as having pro-aging effects.
Compared to how many with anti-aging effects?
Only 78. And that result was highly statistically significant.
Let's just pause on that for a second, because If you're taking one of those 136 drugs, maybe for some chronic condition, you might be getting relief for your symptoms,
Right.
but you could be accelerating your biological clock as a hidden side effect. That's a huge implication for long-term prescription safety.
It is. It means that while these drugs are doing their specific job, they might have systemic long-term molecular consequences that were just never measured during the approval process. We can now ask that question. Does this drug increase my risk of future frailty?
That alone makes the whole project worthwhile. Okay, let's turn to the most exciting part, the proof of concept. The AI found a high-scoring, novel candidate, and they tested it.
They did. That candidate was ouabain.
Ouabain, an old-school cardiac glycoside, a heart drug. It was flagged by the AI with a composite score of 123, but it had basically zero history in broad longevity research.
That's right. So based on that high novel score, the researchers took the next step. They ran the experiment. They treated aged male mice. These are old mice, 20 to 23 months old, intermittently with ouabain for 3 months.
And what did the molecular clock say after those 3 months?
The results were clear. Ouabain significantly reduced their transcriptomic age, their TAGE, just as the AI had predicted.
And did they figure out the mechanism?
One top contributing gene, NREP, that's the neuronal regeneration-related protein.
And connecting back to that RGE system the AI used, why is NREP interesting?
Because NREP has been implicated in major rejuvenation studies before, specifically in heterochronic parabiosis, that famous experiment where you link the circulatory systems of an old and a young mouse.
So ouabain is hitting the same target that we know is involved in genuine rejuvenation pathways.
That's the conclusion, yes.
But anti-aging has to be about more than just molecular markers. Did the drug actually make the old mice physically better?
It did. And most notably, it halted frailty. They used a frailty index, which is a cumulative score of health deficits. Over the 3 months, the control mice got predictably older and frailer. Their FI score went up.
And the ouabain mice?
The ouabain-treated mice showed no increase in their frailty index. The treatment successfully stopped the progression of frailty in its tracks.
That is a real tangible outcome. What kind of specific improvements did they see?
Well, they saw things like reduced piloerection. That's when the coat is all bristled, a sign of poor health in a mouse. They also saw a preserved menace reflex.
For listeners who haven't examined aging mice, what does a preserved menace reflex actually tell us?
It's a measure of cognitive and sensory function. A healthy mouse will blink or flinch if you move your hand quickly towards its face. Losing that reflex means cognitive decline. Preserving it suggests ouabain was protecting brain function.
And given its history as a heart drug, did it affect the heart?
It did. As you'd expect, the treated mice had significantly higher cardiac output compared to the controls. So it helped the heart and slowed down the overall aging trajectory.
One of the biggest hallmarks of aging is chronic inflammation, especially neuroinflammation in the brain. Anything there?
Yes, a beautiful result. They looked at microglia cells in the hippocampus, the brain's memory center. In the ouabain-treated mice, the microglia had a healthier, more branched shape, which is a sign of lower neuroinflammation.
So to recap this whole incredible journey, ClockBase Agent sifted through decades of dusty, forgotten data, used a team of validated AI specialists to spot a hidden signal in an old heart drug.
Right.
And then that drug was experimentally proven to halt frailty, protect the heart, and reduce brain inflammation in old mice.
It's a complete paradigm shift. This transforms all that archived, fragmented data into an autonomous discovery engine. It moves science way beyond the limits of what a single lab can focus on.
And the fact that the findings line up so well with what we already know, the concordance with other databases, hitting known aging pathways, it just confirms that the AI is finding real biological patterns, not just noise.
It really establishes a framework for the future of precision longevity medicine. You can now query how any intervention affects biological age across different tissues and contexts. It's incredibly powerful.
It feels like a fundamental shift away from the slow, traditional one hypothesis at a time model. We're moving toward a future where AI is continuously scanning all available data to find the next breakthrough.
That's it, exactly. It's a precedent not just for aging research, but for all of biological discovery.
We have uncovered some just monumental findings today. We learned that for longevity, genetically taking things away is often better than adding them. We learned that most FDA-approved drugs might actually be accelerating biological aging as a side effect, and that an obscure heart drug, ouabain, is a powerful new geroprotector.
And that leads us to our final provocative thought for you to consider. given that fundamental biological asymmetry, that it's easier to break a system than to improve it, and the significant finding that most FDA drugs may accelerate aging. What new criteria should we demand in future drug development? We need to ensure new interventions promote health span, not accelerate decline as some hidden side effect. And that is an important question for you to explore.